A mutation is simply an alteration in the nucleotide sequence of an organism's genome. Large-scale mutations result in changes to whole chromosomes, which is why they are known as chromosomal mutations. But today, we will discuss mutations that produce changes on the scale of genes. You will explore various types of DNA alterations, as well as their causes. The first way we can categorize mutations is by looking at the effects of a certain mutation on an organism. A gain-of-function mutation creates a gene product that acquires a new or enhanced function, which may be due to an alteration in the amino acid sequence of a protein itself, or due to a change in the regulatory region of the gene that leads to abnormal expression or overexpression of this gene. Typically, gain-of-function mutations are dominant. Loss-of-function mutations reduce or completely eliminate the function of a gene product. These mutations are usually recessive. Eukaryotic genomes consist of much more non-coding DNA than coding DNA. That is why neutral mutations are more frequent than any other type of mutations. They do not influence the phenotype, which indicates that this mutation is located in a non-functional part of the genome. However, some mutations that disrupt essential processes can be incompatible with life. These are known as lethal mutations. It is more important for us to know the molecular basis of a certain mutation. Gene mutations can fall into one of the two main categories based on their effects on the genetic code. A point mutation, also known as a base substitution, involves the alteration of a single nucleotide base in the DNA sequence. Since the genetic code is encoded in triplets, replacing one nucleotide with another affects the reading of a single codon. Point mutations can be divided into three types. A nonsense mutation changes a triplet into one of the stop codons, causing a premature termination of translation. The presence of the new stop codon results in the production of a truncated protein which is likely non-functional. A change in a codon caused by a silent mutation results in no change in the amino acid encoded by it. The reason for this is the redundancy of the genetic code, meaning that the same amino acid can be encoded by different combinations of triplets. Take leucine, for example. Leucine can be encoded by 6 different mRNA codons. So, if a mutation changes the codon CUA to CUG, the amino acid leucine will still be incorporated in the protein at the same position, and overall structure and the function of the protein will remain unchanged. A missense mutation results in a change in the amino acid encoded by a codon. If the amino acid that replaces the initial one has similar properties to the one replaced, this is called a conservative missense mutation. For example, if the polar amino acid tyrosine is replaced by another polar amino acid like cysteine, the mutation is considered conservative. In other cases, where the new amino acid has different properties, it is called a non-conservative missense mutation. The second major type of change in the DNA sequence is a frameshift mutation. This type of mutation is caused by the loss or addition of one or more nucleotides within a gene, which in turn alters the triplet reading frame during translation. Let's have a look at an example that is quite easy to understand. Consider the sentence made up of 3-letter words. Now, if we insert or delete a letter at any place in the sentence while reading it as a sequence of 3-letter words, the part that follows the change will no longer make sense to us. The same thing happens inside the gene. Frameshift mutations can often lead to non-functional and shortened gene products. Since a gene can consist of tens of thousands of base pairs, there is a high chance that sooner or later a frameshift mutation will introduce a premature stop codon. Here is the summary of all the mutations we've looked at today. I really hope my video helped you, and if it did, please like it and leave a comment. And I'll see you in the next one.